I'm happy to tell you about the functional organization of human high-level visual cortex. This is an outcome of a research that took us more than 10 years and involved many collaborators, including Rafi Malach from the Weizmann Institute of Science, Nancy Kanwisher from MIT, Joseph Parvizi from Stanford, and of course many students and postdocs in my lab, foremost Kevin Weiner, Goli Zhigulari, Nick Davydenko, Corentin Jacques, and Nathan Woodhoft. What we're interested in understanding is what are the sequences of brain processes that are involved in visual recognition. In a nutshell, the problem is the following. There's an object in the world, let's say this bird. Photons from this object bounce and hit your eye and form an image, upside-down image on your retina. This information gets transferred to the back of the brain, to primary visual cortex, and then through a series of computation, what constitutes the ventral stream going from the occipital lobe to the temporal lobe, a percept emerge. For example, this is a bird. Much of our research is concerned in understanding the structure and function of these high-level visual areas that are involved in visual recognition. So what's the plan? First, we're going to define these high-level visual areas and describe some of their functional characteristics, and then I'll describe some recent results from our lab in which we improved our measurements using high-resolution fMRI, and we revealed the basic organizational principles of this cortical expanse that have important applications and theoretical implications. What is high-level visual cortex? High-level visual cortex includes the constellation regions in the lateral side of the occipital lobe or lateral occipital temporal cortex and regions in the ventral occipital temporal cortex. This constitutes a constellation of regions that show selectivity to specific types of stimuli. For example, the regions shown here in red, named also the fusiform phase selective area, responds more strongly to phases over other stimuli. In contrast, this region that is about 2 centimeters more medial to it, called the pericarpal place area, responds more to scenes and places and other stimuli. Interestingly, next to phase-selective regions, there are regions that seem to respond selectively to body parts like hands or legs. And there are other regions more posteriorly, especially on the lateral aspect of the occipital lobe, that respond more generally to many objects in shape over patterns or scrambled shape, which constitute lateral occipital cortex. I will illustrate the functional selectivity of these regions with these phase-selective regions on the fusiform. This is a time course from the fMRI during an experiment in which subjects view different kinds of stimuli shown either in the top or on the bottom. The white regions indicate times in the experiment where no visual stimulus was shown. You can see that when visual stimuli are shown, there is an increase in the signal, but you can see a massive increase in the signal when people are shown faces compared to many other stimuli, such as scenes, objects, and even other animate categories. Especially the lab of Nancy Kanwisher has been doing a lot of research characterizing these regions. This is a result from her experiment showing the kind of stimuli that the fusiform face area responds to and the kind of stimuli that doesn't. And basically we characterize these regions as high level because they respond to many kinds of faces, irrespective of format. For example, this region responds to both photographs of faces as well as cartoon and abstract versions of faces, and even to animal faces. However, it is selective in that it doesn't respond to other body parts like hands or headless bodies, and again, doesn't respond to many kinds of objects in building. Of course, there are many differences between faces and non-faces, and one can wonder whether it's really a high-level difference in terms of our percept or some low-level difference between faces and other stuff. For example, faces are round and body parts are elongated. However, other kinds of research suggest that this response is not driven by low-level stimuli. This is an example from our research in which we created silhouette stimuli, shown here in the bottom, either representing profiles of faces, shapes, or scrambled versions of these shapes. Importantly, we control low-level differences across stimuli in the size, shape, contrast, and area of the stimuli. And as you can see, the responses to faces is about double the response of shapes, and even higher as in scrambled objects. Interestingly, the selectivity is maintained if you compare responses to upright face silhouettes compared to upside-down face silhouettes, and this is because when the silhouettes are presented upside down, they're no longer perceived as faces. And indeed, in another experiment, we parametrically varied how face-like these silhouettes were perceived, where 5 shows the most face-like stimulus and 1 shows the least face-like stimulus. And you can see that the response in the fusiform face area increases the more subjects perceive the stimulus to be face-like. So these kind of data suggest that this response to faces is across many kinds of stimuli and not driven by low-level differences across stimuli. Another kind of question you could ask about representation of faces is how sensitive these representations are to metric or image transformation of faces. For example, are these representations sensitive to where I put the picture of a face in the visual field? What view is a face, if it's front or side view, or what size of face I show it. The problem is that in fMRI voxels, we have quite a few neurons, in the order of about 100,000 of neurons. And it's conceivable that there will be different kinds of face-selective neurons within a face-selective voxel, for example, some that like big faces and some that like small faces. So in order to probe face representations, we need to do something a little bit more sophisticated. So one of the things that we did is we used a method of fMRI adaptation to probe face representations. The idea is the following. You can tag specific neural populations in the voxels by adapting them to repeated stimuli. So it's well known from electrophysiology that if you show a face-selective neuron a face, you'll get a nice response. And consequently, if you measure it with fMRI, you get a nice fMRI signal. However, if you repeat the same stimulus again, the neuron will adapt to that face and that its response will be lower, and consequently you'll get lower fMRI signal or fMRI adaptation. Interestingly, this adaptation is very specific. It's not a general habituation of the cell, because if you show a different stimulus, for example, a different face, you'll get a recovery of the signal to its initial state, and consequently a recovery of the fMRI signal. And you could use this phenomena to probe the representation of faces. How would you do that? You'll adapt the neurons with identical faces just to measure the adaptation, and then you could change the size of the face. If the response remains adapted, you'd conclude that the neurons are insensitive to change in the size. However, if the response rebounds, then you'd say that the neurons are sensitive to the transformation, same way that they were sensitive to the identity of the face. So this is the results of these experiments, and basically we show the ratio of response to the repeated stimuli compared to the different stimuli. When you show the same face repeatedly, again you get about a reduction of about 50%. The ratio is less than half. What happens when you show stimuli of the same person but in different position or different sizes in the visual field? You still get some level of adaptation. The ratio is less than 1, but it's higher than when you show the identical faces. And when you show the same face in different illuminations or same faces in different viewpoint, you get a recovery from adaptation. And these data show that there's a differential invariance to face transformation. To some extent, there's some invariance or some generalization across size and position, but the representation is quite specific, for example, when you think about views of faces. So we so far have showed you that the representations, how faces are represented, but the question is, are these regions really correlated with face perception. And to test this hypothesis, we use a paradigm called a masking paradigm, in which we show stimuli very briefly in masking. And the reason that we do this is that because the stimuli are presented briefly, it's hard to recognize them. And then we could ask what happens in the trials when a stimulus is presented and recognized compared to a trial when a stimulus is presented and not recognized. And this looks like this. So I flashed a bunch of images, and you could see sometimes the face and sometimes you couldn't. And the reason is that they're presented very briefly, and sometimes you can pick it up and sometimes you don't. Different people might pick up different stimuli. And basically, the presentation is identical in all cases, and because it's on recognition threshold, sometimes you see it and sometimes you don't. So if we measure the response now from, again, the fusiform face selective regions, we can see that the level of response is tied to the percept. When a face is presented and identified, for example, I recognize Barack Obama, I get a high response from the FFA. However, when a face is presented and detected, this is a blue line, you get a still significant response, but not as high as when it's identified. And importantly, when you present a face and people don't detect that the face is presented there, you get very little response over the FFA. So the level of response over the FFA is correlated with what subjects perceive. Importantly, this is not happening all over the brain. It's not. In fact, if you look at V1, with primary visual cortex receives input from the retina, you can see that under all situations you get a strong visual response, and the same for an intermediate visual area like V4. So it's really these high-level visual areas that are coupled to perception, whereas the low-level visual areas are more coupled to the physical presentation of the stimulus. So far what have I shown you? I've described these high-level visual regions of the human brain, and that they are characterized by selective responses to particular stimuli over others. I've further shown you that the selectivity is maintained when controlling for low-level aspects of the stimulus, and they generalize across formats and to some degree size and position, but to a lesser degree for viewer and illumination. And critically, activation in these regions is tied to percept. So all this is good and interesting, but what I want to convince you is that as a field we want to move to a more precise and quantitative model of high-level visual cortex. So one of the issues is that the prevailing model of high-level visual cortex is something like this. Basically, this is a picture from Nancy Kanwisher review in PNAS from 2 years ago. Basically, the way she described high-level visual cortex is modules, and these modules are specialized for particular domains like faces or words or places. And the main issue is that there's a very limited number of modules, and they're roughly placed in particular location like the fusiform gyrus or the parahippocampal gyrus. And this is, a nice idea, but the main problem is that the data doesn't look quite as simple as it's depicted in these kinds of reviews. So the first problem is that the way the fusiform face area is described like a singular module in the brain, but in fact, if you look at the original paper by Nancy Kanwisher herself, you can see that there are 3 face-selective foci in the fusiform, and she pointed to this one as FFA because she decided that this one is the most reproducible across subjects, and you can see the multiplicity of these face-selective activation also in a more recent paper by Doris Tsao. Now, the problem is that the model says that there's one face-selective model in the fusiform, but different labs use different approaches to deal with this multiplicity, where sometimes they will split the FFA into two patches, like FFA1 and FFA2, like in the paper by Pinkston et al. And this is a paper from my work with Nancy Kenwisher, where we see the same two patches, and we call this the FFA. And this variability is further exacerbated when different groups do different kinds of processing. For example, if they spatially smooth the data, suddenly the FFA is this humongous thing, and then if they do a group analysis, basically half the temporal lobe is the FFA and the other half of the temporal lobe is the PPA. So there are a couple of problems. One is that if you look across the literature, even though the FFA is described as a module for processing faces in fusiform gyrus, the way it appears across the literature is very, very variable. In fact, in a paper by Rainer Goebel and Martin Frost, they claim brain areas. So the question is this a bug or a feature, meaning is this a feature of high-level visual cortex that's just much more variable than, let's say, early visual cortex like V1, or is this a bug that we don't have a good enough model for high-level visual cortex and different people do different things and therefore it appears very variable? And to address this question, we performed a series of experiments in which we improve the measurements using high-resolution functional MRI. Standard fMRI uses voxels of 3 to 6 millimeters, and in our experiments we use voxels of 1.5 millimeters. So what happens in these experiments? Subjects sit in the scanner and they view different kinds of experiments. For example, experiments where they'll see faces and body parts and objects to map these high-level visual areas, and in addition, they participated in other more standard experiments to map the visual field and localize other regions of selectivity. And we improve the measurements in several ways. One, we used higher resolution fMRI. Second of all, we used multiple measurements in the same subjects. We didn't spatial smooth our data. We did the analysis on every single subject. And we use cortical surface visualization because it gives us a really nice view of what happens across the brain, and we paid attention to anatomical landmarks. So what did we find? What we found is that the high-level visual cortex is much more organized than previously thought, and specifically there are 4 principles that can describe this organization. The first point is that there is a co-localization between functional regions and anatomical landmarks. So on the left you can see the ventral view of a right hemisphere in which I marked the anatomical landmark. So the fusiform gyrus is shown here in pink. It's bounded on the right medial side by the collateral sulcus and on the lateral side by a different sulcus called the occipital-temporal sulcus, and there's a little sulcus that runs in the middle of the fusiform gyrus called the mid-fusiform sulcus. If we look at this is an anatomical map of the ventral stream. If you look at the map of face selectivity, these are regions that respond more to faces than other objects. You can see 3 patches of face selectivity, 2 on the fusiform and 1 in the inferior occipital gyrus. And notably, these regions co-localize with anatomical landmark. Specifically, one region that we call the mid-fusiform faces, M-fus faces, is coupled with the mid-fusiform sulcus. The second region is about 1.5 centimeters more posteriorly, and it straddles the fusiform gyrus and the OTS. And the third region is outside the fusiform gyrus and in fact still in the occipital lobe. You might wonder how good is anatomy as a predictor of functional region, because isn't there a lot of intersubject variability in the anatomical folding of on top of the variability in the activation, as we've just shown before. For example, if you look at a citation from David Van Essen, he says, comparison of results obtained in different individuals is hampered by the dramatic variability in the pattern of human cortical convolution and in the location of cortical areas relative to these folds. Additional confounds arise from the complexity and variability of human cortical convolution and from individual variability in the size, shape, and location of each cortical area. So the prevailing view in the field is that there's a lot of variability and anatomy is not a good predictor. However, I think this view is based on kind of like some notions based on some measurements that people did with scanners 10 years ago, and today with better measurements we could actually show that there's a really nice correspondence. In fact, we wanted to see how variable is anatomy and how good is anatomy a predictor of functional activation. So first of all, if you consider the mood fusiform sulcus, it always divides the fusiform into lateral and medial portions, and you can find it in 97% of the hemispheres. And you could ask, how good is this as a predictor of function? And in fact, in 83% of hemisphere, and we're talking about 83 out of 38 hemispheres, if you look for the anterior tip of this mid-fusiform sulcus, it's going to align with an anterior tip of the functional activation, the emphus faces. And also, if you consider the lateral versus medial aspects of the fusiform gyrus, the mid-fusiform faces is on the lateral aspect of the fusiform gyrus in 89% of the hemispheres, and when it extends to the occipital temporal sulcus and rarely into the middle aspect of the fusiform gyrus. So basically what I'd like to say that there is really a nice coupling between function and anatomy, and this is clear when you look at individual subjects and when you start doing metric measurements. The second principle is a principle of a global topology. What that means that there's a consistent spatial arrangement between different functional regions. So I'm adding to the functional maps of phase selectivity, again shown here in red. Now in green are regions that show selectivity for limbs. And interestingly, between phase selective patches, we tend to find a limb selective patch. For example, between the emphus faces and the epiphyse faces, consistently you'll find a body part selective patch, which is between these two patches and more lateral to them on the occipital temporal sulcus. And this global topology is not only a feature of the ventral surface, it's a more general principle of organization of face and limb selectivity. So these are just 4 other examples. These are 4 brains, and you can see that in each of these brains, the limb selective activation basically falls in between the 2 phase selective patches, the emphus and epiphyse faces. And as I said before, this organization extends onto the lateral surface. So before I showed you mainly stuff on the ventral surface, which was here. But now I'm going to show you stuff also on the lateral surface. And what I want to point your attention to is that between 2 phase selective patches, you'll find a body part selective patch, and this is true also for the lateral surface. You have 2 phase selective patches, and between them falls a body part selective patches, or conversely, between 2 body part selective patches, you have a phase selective organization. So you have this alternating patterns in face and limb selectivity across ventral and lateral temporal cortex, and in fact it generates a very nice and consistent organization across subjects. This multiplicity of activation and this alternating organization or this global topology between faces and body part might reflect their actual topology in the real world, where faces are usually attached to bodies and they have this regular organization in the world. So it might be that this topology has been mapped to the brain. So this the schematic on the right summarizes this organization, where you have two face selective patches on the fusiform, the M-fus faces and the P-fus faces that are separated by a limb selective patch, the OTS limbs, and then in the lateral surface surrounding an anatomical structure called the posterior infratemporal sulcus, you'll find three limb selective regions on again distinct anatomical locations: the lateral occipital sulcus, infratemporal gyrus, and middle temporal gyrus, and these are separated by two face selective patches, one on the inferior occipital gyrus and another on the posterior aspect of the superior temporal sulcus. Interestingly, this global topology of face and limb selectivity is not restricted just to arrangement of faces and body parts, but it also extends to other functional areas. So you might notice that here in the ITS, there is a region that seems to be devoid of functional activation, and you might wonder why is this happening. So what I'd like to point out, it's devoid of face and limb selective activation, but it's not really devoid of activation. And if you'd actually run an experiment where you show moving versus stationary stimuli, you'd find a region here that's motion selective, and that it responds more to moving stimuli compared to stationary stimuli. And this just shows this global topology and consistent arrangement of faces of activation in high-level visual cortex extend to many functional domains, and not only to domains related to the human body. So the third principle, so the first principle is anatomy, the second principle was global topology. The third principle is that this spatial relationship between functional regions extends to retinotopic maps. So one way to map the human brain is by mapping the maps of the visual field, and in fact visual field maps have been discovered throughout the brain, and it turns out that even though these face selective regions are not retinotopic by nature, they're insensitive to position, they have a consistent spatial relationship or congruence with retinotopic maps. For example, the inferior occipital gyrus or IOG faces is always lateral to a visual field map called V4. The P-fus faces is lateral to the VO maps, and the M-fus faces is lateral to the PHC maps. So this global topology also extends to relationship to retinotopic maps. And the fourth principle is that of an organization is that we want to mark regions only if their functional characteristics are stable across time and task. So in this example, we've measured the face selectivity in the same regions across 4 experiments, across 3 additional experiments, and 2 of them were done 5 months later. And what we varied across experiment was a design as well as a task. Sometimes they did a categorization test, and sometimes they did just pressing a button, 1-2 images repeated. And what you should take note is that this is a response from this region, from the M-fus faces, you can see that the response across all experiments is highest for faces and shows the second best response to limbs, and then a lower response to other categories. So the profile of response as well as the selectivity is maintained over time and task. So basically these findings reveal that high-level visual cortex is much less variable than depicted in the literature, and if you use high-resolution fMRI and multiple measurements and pay attention to anatomical landmarks, you can increase the accuracy of determining regions, and you can determine them on a single subject basis. And moreover, this kind of model provides a detailed model that describes organization at the level of a single subject. So the way we like to think about high-level visual cortex is that it's not made of these discontinuous modules that have no relation to each other, but in fact, the cortical expanse of high-level visual cortex is covered by multiple maps that have a consistent relationship to each other and produces interrelated topology. And in fact, in a given region, you could have more than one kind of functional map. For example, you could have a map of face selectivity as well as a map of the visual field. And we think that the concept that best describes this organization is the concept of cortical tiling. So basically, in a given anatomical expanse, like the ventral temporal cortex, you could have multiple maps of retinotopy, or object selectivity, or place selectivity, or face selectivity, or body part selectivity. Each of this map produces some covering of this visual space, but the combination of all of these maps produces the styling of the cortex, where some of these maps converge and some of these maps diverge from one another. And we think that the fact that some of these maps converge and diverge might have functional implications, because this converging and diverging pattern can increase the repertoire flexibility and efficiency of visual processing. So different amounts of spatial convergence and divergence can enable factorial combinations of multidimensional information. So for example, you could combine faces and body parts, or body parts to have an understanding of the human body, or you could combine representation of body parts like hands with objects to understand how you manipulate how these objects interact. Second of all, the fact that you have multiple representations and different levels of convergence and divergence produces some flexibility, because maybe sometimes you want to access one kind of information, or just information about the objects or the hand, and sometimes you might want to access the combined information, like how does the hand manipulate the objects. And finally, the fact that we have a multiplicity of regions that are selective for a given stimulus might be an implementation of parallel processing in the brain, in which this increases in efficiency because you can process different aspects of the stimulus in different regions. For example, you might process identity information about faces in one region and emotional information about faces in another region, and splitting the task into smaller tasks in different parts of the brain might make the processing faster and more efficient. So not only that this organization, we understand it much better, but because now we have a model that is applicable in the single subject level, this has a lot of clinical application. For example, we had a patient that was a candidate for an epilepsy surgery, and one of the things that the surgeon does is test functional selectivity in subject brain, because if you would find that the epilepsy is on a face selective region, you might not want to remove it, because that might disproportionately impair face processing in that subject. So this is an example brain of one of our patients, and just to remind you, this is a fusiform gyrus of this patient, and this is a mid-fusiform sulcus. And if you've been paying attention to me before, our mid-fusiform activation should be close to and lateral to this sulcus. And if you measure, and then there's another focus of face selective focus more posterior to that. And if you measure as electrical signals from these electrodes are electrodes that are implanted subdurally before the subject's operation, you can measure the electrical signals, and this is the electrical signal over time from these two electrodes as the subject is viewing faces or limbs or cars or houses, and you can see a much stronger increase in response to faces compared to the other stimuli in both electrodes, and you can see that this increase in response happens in about 100 to 300 milliseconds after the stimulus is presented. And importantly, we did fMRI on the subject, and we can show that these electrodes are exactly where the fMRI shows in the subject. This is fMRI of the subject, the mid-fusiform face patch, and the posterior fusiform face patch. And the subject has the same topology as we saw on other subjects. For example, the IOG patch is lateral to his V4, and the PFUS patch is lateral to the VO maps, and the MFUS patch is anterior to these maps. So basically we find a really nice correspondence between different kinds of measurements, not only fMRI but also ECOG, and we can see that if we know something about anatomy, we really have a really nice predictive model of where these activations should land, and it actually works on a single subject basis. One of the things that we were able to do on a patient that we cannot do on a subject is to see what happened if we stimulate these electrodes, and basically when surgeon is neurologist, Dr. Bravisi drives current in these electrodes, whether it would impair face processing or not, and if this is specifically for faces. So in the experiment I'm going to show you is this video. The doctor is going to be talking to the patients while he's looking at him, and he's either going to drive currents into the electrode, you'll see the current amplitude, or he's going to do the same kind of thing but not drive any current. We call this sham trials. I'll be this again. Close this again.
Just look at my face and tell me what happens when I do this, alright? 1, 2, 3. Nothing? Okay. I'm going to do it one more time. Look at my face. 1, 2, 3.
You just turned into somebody else.
Tell me
Your
about
face metamorphosed. Your nose got saggy, you went to the left. You almost looked like somebody I'd seen before, but somebody different. That was a trip.
Could you see my eyes in place?
I could see your eyes, but you could have been somebody else who had, well, he has similar eyes to Dr. Bravisi, but you were someone else. Your whole face just sort of metamorphosed.
Did it get warped?
Yes.
Did it keep its own features?
No, it's
Or
almost
shape?
like the shape of your face, your features drooped. Yeah, sort of like they drooped to a particular direction, kind of stretched and drooped.
All right. Ready? 1, 2, 3.
Yeah, it metamorphosed again, and you look like someone I've seen before, but maybe a different person in my memory. Almost like your nose kind of shifted to the left a little bit, and your look just changed.
It did.
Yeah.
Tell me more.
Well, I mean, I don't have a photographic memory, but it just, when you turn into something.
Ready? Tell me if the same thing happens again. 1, 2, 3.
Nothing happened.
Nothing happened. Are you sure?
Sure.
How about now? 1, 2, 3. Okay.
Yeah.
The same thing happens?
The bottom of her face sort of metamorphosized up, kind of stretched up to give her a different look. It wasn't pretty.
Anything change?
Something did change for a glitch.
What did you feel?
It felt like the TV moved and then the balloon moved to a certain extent.
I see.
But it wasn't very clear. It didn't stay there. Just
Got it. Could you still understand if it's the TV?
and it happened so fast. It's just something different happened.
Did the shape of the TV change?
Not necessarily. Almost like it just shifted a little bit.
Okay. Read again.
Hope you're feeling better.
Okay. How about now? 1, 2, 3.
Hope you're feeling better.
Did anything change?
Very slight when I first read the word hope. I know it doesn't describe a whole lot.
It says you were talking about. Okay? Now?
No.
I'm not feeling it? How about now?
Yes. Your face started to skew to the right and protrude.
Okay. Perfect. Thank you.
So basically, this movie shows that by stimulating these electrodes on the fusiform NFUs and PFUs faces, you get a very specific distortion in face perception. This happens when he's stimulating, not when he's putting sham. And even though he's documented that maybe something is a little bit shifting, the fact for when he's looking at the TV or the balloon, the fact that distortion is very minimal is quite striking, and basically this really shows us. So, guys, in the second part of the talk, I revealed, like with better measurements, we have an organization framework of functional region, and this organizational framework in high-level visual cortex consists of 4 principles: congruence between a functional region and an anatomical landmark, that functional regions are arranged in a consistent global topology, and that they're congruent with visual field representation, and these characteristics of these functional regions are consistent over time. Important organizational frameworks explains this high-level visual cortex, where each region is a component of a larger principled map, as opposed to a series of discontinuous and unrelated modules. Define functional regions from anatomy and a topological model, do cytoarchitectonics and connection patterns along regions constrain this organization, and do these organization principles extend beyond visual cortex and to other species. Thank you very much for your attention.